All right, we're going to get started. So, our next and final presentation is titled EVO, a foundation model for generative genomics. And this team is creating a model of the human genome to advance our understanding of DNA and the implications for human disease. So, let's welcome our final team.
Cool. Hi, everyone. I'm excited to share the progress that our team has been making over the past year. We have been making a lot of progress on developing biological foundation models, and these are models that are trained on genomic sequences. But hopefully we'll see by the end of this talk that even though we only train on DNA, these models can actually generalize and do quite a lot. And to begin, I just wanted to bring up that today we've been talking about human-centered AI, and actually a lot of the most pressing issues that humanity faces are actually biological problems. So humans are biological organisms, and so health and disease are biological processes. Biological processes underpin many of the problems with food security and agriculture, and biological processes also affect things on a global scale in terms of climate and sustainability. And a lot of the problems of why these problems seem so intractable is that right now it's actually very hard to control biology and kind of use it for human good. I wanted to start with this quote by Frances Arnold, where she says, "Today we can, for all practical purposes, read, write, and edit any sequence of DNA, but we cannot compose it." And so there's been tremendous technological advances in technologies for genome sequencing and genome synthesis and genome editing, but it's still very difficult for us to compose completely new sequences of DNA, for example, to control biology in ways that we would want. And part of the reason for this is that, if you think about it, the biological genetic code is completely unintuitive to us as humans. So the DNA sequence is this really long sequence of 4 base pairs, A, C, G, T. The human genome is 3 billion base pairs long. I look at DNA sequence almost every day, and I probably still couldn't tell the difference between random DNA sequence and the sequence that comes from an actual organism. But there is information in these sequences, and we have been using AI tools to be able to extract this information from the sequence and learn from it and design new DNA sequences from that. So the tools that we've been using have actually been inspired by these algorithms that have been making tremendous progress in learning higher level rules from sequence information alone. So for example, these models are trained with very simple tasks like predicting the next token in a sequence of tokens, where a token could be an English word, for example, or a token could be a DNA base pair. And the idea is that even though predicting the next word in a sequence of words seems like a very simple task, potentially, if you train a model on a sufficiently complicated data set, then next word prediction in language actually encompasses a lot of very complicated tasks within it. One way that people like to think about these large language models is that even though these models are not learning an explicit world model, the world has left enough of an imprint on text from the internet, for example, so that these models can simulate some aspects of the world from learning the sequence patterns alone. And so in biology, we typically like to think of these 2 general domains. We have genotype, which is the DNA sequence, and the sequence encodes for phenotype via some complex biological mechanism, where phenotypes are the biological physical traits that you observe, for example, and it's often these traits and these phenotypes that we're actually interested in biology. But it's also often very difficult to collect lots of data to train large models at the level of phenotypes in biology. But it's been very easy to collect lots of data at the level of DNA sequence. And actually, the amount of DNA sequencing data outpaces data generation on the internet. So potentially, we might be able to train models on DNA sequence to get information about phenotype and mechanism. And this is because, even though evolution acts on physical traits in the phenotypes, the results of this evolution get written back into genotypes via natural selection. So in the same way that the world has left an imprint on text from the internet, the world has also left an imprint on DNA sequence through evolution. So an intriguing hypothesis would be that maybe if we trained a model with next base pair prediction, this seemingly simple task could still teach the model aspects of very complex biological processes like mutations that are correlated with health or disease, aspects of protein biochemistry or RNA structure, or maybe even higher level systems biology. This hypothesis has actually been pretty productive for a class of models called protein language models. So instead of training on DNA sequences, these models are trained on sequences of amino acids that make proteins. Again, using simple tasks like predicting the next amino acid in a sequence. And suffice to say that models trained on large databases of protein sequences and structures have revolutionized the study of molecules. So, for example, the Nobel Prize in Chemistry in 2024 was awarded to AI systems for structure prediction and design of proteins. So, we're interested in molecules, but we're also interested in how these molecules can come together to make a complete organism. But when you think about it, even a very simple single-celled bacterial organism is orders of magnitude more complicated than any individual molecule. And we are interested in learning things at this level of complexity using machine learning. How do we even do that? It seems very complicated. Well, it turns out over the past 30 years or so, people have been sequencing the complete genomes of millions of organisms and depositing these sequences in publicly available databases. And in theory, embedded in all this information is the information required to create whole organisms. So what happens if we actually train, say, language models on this data? So last year, we put out this model that we called EVO, or EVO1 for the sake of this talk. This is a DNA language model, and really all this model is doing, it's doing a very simple task. All this model is doing is you give it a DNA sequence, and it predicts the likelihood of the next base pair in the sequence. But the model that we use for doing this is actually very large and complicated. The model is a neural network that has 7 billion parameters. It's trained on 300 billion base pairs of DNA sequences. The model of EVO1 is actually trained on organisms called prokaryotes. So these are archaea and bacteria, very simple organisms with very simple genomes. But we did this we restricted to this data for EVO1 because these genomes are a lot less complicated than the genomes of say, humans. What we wanted to get from this first proof of concept is two key ideas. So the first key idea is that even though we're only training a model on DNA, because of basically the beauty of how biology stores information, actually this DNA sequence directly encodes for RNA sequence, and this RNA sequence also directly encodes for protein sequences as well. So it's kind of like a hack. Even though we're only training on DNA sequence, we potentially get RNA and protein information for free out of the model. But also, we train these models to process an input sequence of 130,000 DNA base pairs. So at this scale, we not only get information about individual molecules and individual genes, but how these genes interact with the broader context of multi-gene systems, say, or even complete genomes. Part of the reason why we trained a model at 7 billion parameters on 300 billion tokens is that we observed this phenomenon that's also been observed in AI models of language and vision, which is basically as you increase the amount of compute used to train the model's ability to predict the next token gets better, really predictively, actually. And so scale is one dimension with which you can use to improve language modeling performance. Another dimension is the architecture, or the architecture of the neural network. So we actually use this new architecture called striped hyena that has favorable scaling compared to the traditional transformer architecture that has been used a lot in natural language. So once you have a DNA language model, what can you actually do with this thing? One thing that we did with this model is we used it to simulate what happens if you make a change to DNA sequence in an organism. How will that DNA sequence change affect the organism's fitness or its viability, its ability to reproduce? So we actually ran this very interesting experiment where we made what's called a premature stop codon insertion. It's actually a very bad mutation. If you put this mutation into a gene of a natural organism, it would make sure that gene doesn't code for protein, so it basically knocks out the gene. And interestingly, when we made a premature stop codon insertion and then fed that sequence to EVO2, if we put that bad mutation into the sequence of an essential gene, the EVO likelihoods actually go down by a lot. So the model really doesn't like those mutations in those positions. But if we put a premature stop codon insertion into a non-essential gene, EVO likelihoods on average don't change all that much. So the model has learned which genes in a genome are more tolerant of these really bad mutations, and perhaps even which genes are more or less essential to an organism. This is useful in certain tasks. If you're developing an antibiotic, for example, you want to target the essential genes for the bacteria rather than the non-essential genes. Another thing that these models can do is similar to how ChatGPT can generate code for you or write an email for you, these models can generate new DNA sequences as well. And so we actually specialize the EVO models to generate these CRISPR-Cas sequences that are used for genome editing. They're actually pretty complicated molecular machines that involve proteins and RNA working together. So we were able to generate a DNA sequence that contained these protein and RNA molecule sequences within them. We can actually then chemically synthesize the DNA and then test if the products encoded by this DNA work in the lab, and actually we observe that they do. So this AI-generated CRISPR-Cas system, EVO-Cas9, actually has comparable in vitro cleavage of double-stranded DNA to SP-Cas9, which is the state-of-the-art system for genome editing. But we also observe that this EVO-generated Cas9 has less than 80% sequence identity to anything in nature. For context, this is enough sequence identity, sequence novelty to break any patents around CRISPR-Cas. So at least in the eyes of the US Patent Office, these molecules are novel. We then wanted to push the novelty even further. And for this, we did a pretty interesting experiment, I think. So we used the fact that the model learns information across multiple genes and that bacterial genomes often have functionally related genes right next to each other on the genome. So we can prompt the model with a gene and then get another gene in the response. So we asked, what happens if we prompt the model to generate genes that in nature have extremely high novelty, trying to elicit novelty or creativity from the model? The particular gene that we generated, these are genes called anti-CRISPRs, so they inhibit CRISPR, and we can prompt with, for example, an anti-CRISPR associated gene. So we prompt the model with DNA and get DNA in the response. We actually find anti-CRISPR genes in the response. So when we test these in the lab, they do have activity against CRISPR-Cas systems. But very interestingly, for a couple of these genes, we then compare their sequences to all the genes that have ever been discovered in biology, not just in the training data, but all the genes ever discovered, and we find no significant sequence similarity to any of these genes, indicating, I think, that the model is capable of generalizing quite a lot in sequence space beyond existing genes. It's actually able to do some amount of non-trivial creativity. We then use the model we prompt it to generate 100 billion base pairs of AI-generated DNA, spanning thousands of putative functions that we make publicly available. We also have the model, instead of generating a single gene, we asked the model to generate 1 million base pairs. And we thought that these sequences would be totally garbage, but actually, along a number of metrics, like the amount of predicted genes on these sequences, they look somewhat realistic. We find tRNAs on these sequences that cover all 20 canonical amino acids. So we kind of liken this to the first versions of these image or video generation models, like Will Smith eating spaghetti or something like that. They would generate these blurry images that were somewhat coherent but clearly wrong. EVO1 is kind of generating these blurry pictures of genomes. So then we wanted to expand this modeling paradigm beyond bacteria and archaea, these prokaryotes, to include a third domain of life, the eukaryotes. And this is a very important domain of life. It includes plants and animals and humans. So we want these models to also be useful for human genetics as well. The problem with eukaryotic genomes is that they're a lot more complicated than prokaryotic genomes. So they're in the billions of base pairs instead of the millions of base pairs. There's a lot of sequences of these genomes that don't code for anything. They have very low information content. So when we were developing this model that we put out a few months ago called EVO2, we trained this model on 9 trillion base pairs spanning everything from bacteria to insects to plants and animals and the human genome. We had to do a lot of work in, especially for the eukaryotic sequences, cleaning up this data and making sure we were training the model on information-rich content, similar to a lot of the data processing pipelines used in natural language model training, actually. We also scaled the model to have to be able to process an input sequence now of 1 million base pairs instead of 100,000. This actually took a lot of engineering and work. We, for example, developed an improved version of our neural network architecture that could have good utilization of the training infrastructure at 1 million base pairs of input context. And this is especially difficult because we also trained the model on with 40 billion parameters now, continuing the scaling laws. So this not only required a lot of architecture development, but also a lot of infrastructure engineering. We partnered with NVIDIA, who donated 2,000 H100s for this training run. Greg Brockman did a sabbatical from OpenAI and helped us train these models. And the result is a model that was trained on with 10 to the 24 floating point operations. For context, this is the largest model by compute flops ever trained in biology. It's also, we make this model fully open, so we open source not only the model weights and the code for running the model, but we also open source the pre-training code that's been battle tested on 2,000 GPUs freely for the community. We open source the pre-training data, and by this metric, this model is one of the largest fully open models across all modalities, including language and vision. So EVO2 is also now available. What can you do with this model? So again, we can try to interpret it by mutating the sequence inputs and measuring the effects on the model's likelihoods, right? Like what we did for the bacterial gene essentiality study. But now we can do this for human genes. So all of humans have some genetic variation, and most of the time these changes in our genomes are actually completely benign and they don't do anything, but in rare cases they do lead to disease, so it's very important to determine which ones lead to disease. We hypothesize that because EVO2 has been trained on a lot of living organisms, that it learns which mutations are compatible with life. And so if you have very high likelihood under EVO2, potentially those mutations are compatible with life and with function and probably with health as well. But if you have very low likelihood of under the model, then potentially those mutations are incompatible with life and might lead to disease. So we actually benchmark the model on this database called ClinVar. It's a database compiled by the NIH on variants that do lead to clinical outcomes for human health. And actually, EVO2 is not specialized as a human clinical variant effect predictor. It was just pre-trained on 9 trillion base pairs across all evolution, but it actually outperforms in many cases models that were specifically developed for variant effect prediction. In some classes of variants, this model is actually state-of-the-art. So especially for these variants that are not single character or single nucleotide substitutions, so these are more complex variants like insertions or deletions or duplications, the model is state-of-the-art and outperforms models that were specifically trained for variant effect prediction in the pre-trained model. So this is very cool to see. The model is also very competitive, and in non-coding variants for this gene, BRCA1, it's also state-of-the-art. BRCA1 is a gene that's often implicated in breast cancer, so we're also very good at predicting pathogenicity in this setting. Another thing you can do with these models is not just interpret them like black boxes, you can actually peer into the neural network parameters. And this is inspired by some of the work done by a company called Anthropic on a language model called Claude. And they found actually portions of the model's internal representations that would fire when it received inputs related to the Golden Gate Bridge in this case. So we did this kind of interpretability for Evo2. The idea behind this is that the model's representation space is a superposition or a combination of different human interpretable concepts. And if you want to disentangle these concepts, you should train a sparse representation. So we train a sparse autoencoder on these representations and see if these sparse features are more interpretable. And we do find that they, in general, are. So for example, we find features learned by the model that only activate when they see non-coding RNAs in the input, or protein level features like alpha helices or beta sheets, or DNA level features like certain parts of the DNA that bind to proteins. One thing that Anthropic found in their interpretability work actually is that there's some features that fire when there's a bug in your Python code, for example. We found a feature that would fire when it saw a frame shift mutation in a gene. So by analogy, this kind of interpretability could find errors in computer code and genetic code as well. Beyond this, Evo2 is also a generative model, and it's able to produce potentially pretty complicated sequence designs. So we wanted to see if now, with a very powerful model, we could realize whole genome design and actually design a complete genome with an AI system. The genomes that we design in this case are the genomes of these things called bacteriophages. So they're very simple viruses that infect bacteria. And these phages are somewhat tractable because even though they're complete genomes, their genomes are much smaller than the genome of a bacteria, for example. But it's still a very complicated design task when you're doing whole genome design. You need to design not only multiple genes, but also the non-coding sequence that regulates those genes, and these genes all interact with each other, and they interact with genes in the host. So another reason why we want to do this beyond its complexity is that phage therapy is actually emerging as a powerful therapeutic modality for multi-drug resistant bacteria. So, for example, in the past few years, this is a very famous case. Tom Patterson almost died because of a multi-drug resistant infection, but his life was saved actually, and he made a full recovery by phage therapy. And here is a photo of his wife holding a picture of the phage that saved his life. So the phage design actually has a lot of potential benefit for therapeutics as well. So we designed variants of this phage called Phi-X174. This is a very well-studied phage in molecular biology. It was actually the first DNA genome ever sequenced back in the 70s. It's the first genome ever synthesized back in 2003. So potentially we also wanted to see if we could make it, or variants of it, the first AI-generated genomes. To do this, we fine-tuned Evo1 and Evo2 on 15,000 examples of phages from the family of phages around Phi-X174. We then generate new genome sequences. We can, again, chemically synthesize them and test them in the lab. And these experiments yielded 16 viable phages. They actually have substantial evolutionary novelty. One of these phages has less than 95% average nucleotide identity to any phage that we've ever discovered. If we discovered a new phage like this from nature, it would technically count as a new species. So we meet the species threshold in terms of our novelty. And we also find that if we pool these phages together into a cocktail or a mixture of the phages, they can rapidly overcome bacterial resistance. So in this case, we have these 3 strains of bacteria that developed resistance against the original phage Phi-X174. But if we mix the AI-generated phages together, then that pool of phages can rapidly overcome resistance on the part of the bacteria. So we're excited by this because there's a lot of diseases that are caused by drug-resistant bacteria. 1 million people die a year from multi-drug resistant bacterial infections. We lose maybe a fifth of crops globally, and there are phages that do target strains of bacteria that are the culprits of these diseases. So potentially with our tools, we can deliver maybe more powerful phage therapies both in the clinic and agriculturally. So hopefully I've convinced you that even though we're training a model with actually a very simple task, all we're doing is we're predicting the next base pair in a sequence. Surprisingly, this modeling paradigm is useful for a broad range of tasks across biology, spanning prediction and design tasks from the scale of genes to genomes. So for example, we can predict how single nucleotide changes affect the function of this key breast cancer gene. We can predict how different regions of the genome encode for these different genetic elements. We can predict how mutations to genes affect whole organism fitness. We can design these anti-CRISPR proteins at the single gene level that are quite novel. We can design these multi-gene systems like CRISPR-Cas that involve protein and RNA. And we can design complete genomes like these phages that are potentially therapeutically useful as well. So I wanted to, again, bring it back to this quote by Frances Arnold. This is actually from her Nobel lecture. And at the end of this quote, she says that evolution does learn how to compose, the code of life. And so how we're seeing this line of research is now we're also learning from evolution on not only how to understand biology, but also hopefully how to manipulate it for the good of humanity. We make we do all our work, we do very open science, we make all our work publicly available, and so at these GitHub links, you'll find links to the training code, the inference code, the model weights, the training data. We also document everything in preprints and publications, so the references for these are on the slide. And I wanted to talk very briefly about what we're interested in working on moving forward. And a lot of this is based on this finding that this pre-trained model, in some cases, is already state-of-the-art at predicting which variants of the human genome lead to disease. This could actually be a very useful technology. So one very low-hanging fruit idea is to also provide the model with human-specific information so that it learns, which variants of the human genome are also more or less associated with disease. We can also give it human-specific functional genomics data as well. And so if we integrate evolution and functional readouts from humans, not only would we be able to potentially get much better prediction of which individual genetic variants lead to disease, but what we're excited about is potentially learning how combinations of these variants also lead to much more complex disease. So why is this important? Well, it's actually been found that for simple genetic diseases, if you actually have strong genetic evidence for a disease, then a drug, for example, is 2.6 times more likely to succeed in clinical trials. This, so far, has only been limited to very simple genetic diseases where we can very easily pinpoint the genetic causes. But if we have a very powerful model that can pinpoint more complex combinations of genetic causes for disease, and if you can, double or triple the amount of cures coming out, then this could potentially be very useful. And the last thing that we're interested in, it's kind of hard to use Evo2 right now. You have to download the weights off of Hugging Face. You have to run them on a GPU cluster with a Python API. So what would it actually look like to make this a lot easier to use for researchers more broadly? What would it look like to develop a graphical interface for generative tools for biologists or people more broadly? Or what would it look like to, say, develop a high-level programming language for generative biology? Cool. So that concludes the talk, but I have a lot of acknowledgments. And so this is work. I'm representing a very large team. Our team is actually incredibly diverse in backgrounds and interests. We have people working on machine learning architecture and infrastructure engineering and training models at scale. We have people working on computational biology and actually evaluating the models and curating the training data. We have people working on experimental biology and testing things in the wet lab that comes out of these models. The Evo2 work itself is also a big collaboration. I mentioned our collaboration with NVIDIA earlier. There's also a big collaboration with Arc Institute. the labs of Patrick Hsu at Berkeley and ARC, Hani Goodarzi at UCSF and ARC. Goodfire is a startup that did a lot of the mech-interp work. Collaborators at Sid Pinglei's group at UW did a lot of the experimental validation. We have a safety and ethics team that also helps us evaluate responsible use of these models here in Stanford Medicine and at Hopkins. And finally, with that, I'll conclude the talk and we can proceed to the Q&A, I guess.
Thank you. I believe you have team members coming up too.
Oh, yeah.
Are they here?
And we have
Okay.
2 team members joining me for the Q&A.
Yes, so you all can take a seat in these chairs. And as before, we'll start with questions from our selection committee.
I had first just a clarification question because it's beyond my knowledge. Why do I want to make an anti-CRISPR gene?
Yeah, so anti-CRISPRs are often used in like molecular circuits that involve CRISPR. So maybe you don't want CRISPR to be modifying all your cells. Maybe you only want it to modify like brain cells or your liver cells and then turn it off in all other cells. So this anti-CRISPR could be a very important component in like a logic system for inhibiting CRISPR in certain cases. You have to couple it with some logic on top. But yeah.
How important was it going from the standard transformer to the striped hyena to enable these results?
So Garrick did a lot of has done a lot of experiments in architecture and convolutions, I think.
Yeah, I think it's a optimization problem. The transformer has been highly optimized for language. The information content per token in human language is much higher than in DNA. Convolutions, like in striped hyena 1 and 2, fit that much better with the locality bias. And so like through repeated experiments, we found much better scaling, both in terms of per step, so per piece of data, how much is your model learning, and also per time. So it trains faster and learns more in that time.
But was it efficiency largely, or did you find that it was a sort of an inferential and learning bottleneck?
Yeah, I think part of it is a learning bottleneck where the transformer just did not learn as well as you might expect. And the gap is quite big per piece of data.
And is there another piece of magic in architecture that we need to get to next?
So there's definitely more work to be done on optimizing architectures for hardware efficiency. So our Evo2 architecture is very efficient at longer sequence lengths. At shorter sequence lengths, there's a bit of a gap to close because there's been so much work to optimize transformers, but actually the team is making progress on that. There's a preprint from a lot of the names that you just saw, including me, that came out today that answers that question.
Oh, I'll go look for it instead of asking you right now. Oh, my other question is the following, and it's in the context of the biosecurity work. To be frank, it sounds like these are afterthoughts right now, frosting on the cake. We have a safety and ethics team, by the way, as opposed to central in this research, we want to consider dual use at every single step, and inadvertent as well as malevolent, making viruses and so on, anti-CRISPRs, not for stopping the therapy at the liver, but for stopping something more essential, like bacterial response to virus. Anyway, so I'm just curious what thinking is about how much, I'll ask it this way, and it's more like this is for all of us as a humanity. When we have a very creative effort like this, that's so promising for finding cures, and it's just amazing. We need to pursue this with vigor. It's very exciting work, and I'd much rather ask questions all about that. But I have to ask, what's the responsibility for the kind of percentage of our efforts we go into as we do this work with thinking through the red teaming and the safeguards and guardrails and policies?
Yeah, I think Steve and I and Samuel have thought a lot about the safety component. It's definitely not an afterthought, and we actually do think about it quite a lot. Well, it's at all the stages. Like for example, before the phage work, we went through, like, we do a lot of our work at ARC Institute for experimental validation. So we make sure we clear it with there's actually a lot of precedent, for example, in biosafety that goes back to the Asilomar conference, that we can somewhat repurposed for this, but I think there needs to be some discussions on should we repurpose some parts of these established protocols for AI-generated systems. So for the phage project, for example, prior to doing any work, we cleared it with Arc Institute. All of the work was done at the relevant biosafety level, but with enhanced precautions as well. So we did all our experiments in a hood, for example. This is not some people just work with phage on the bench, for example. All of our bio waste was put and was disposed as biohazardous
And you
waste.
ruled out mouth pipetting, I think.
Oh yeah, no, I don't know. Samuel snuck in.
Must be a
some
joke.
mouth-
There is no mouth by
and we were very thoughtful. We in all of our papers actually we devote substantial like word real estate to biosafety and bioethics, including the phage paper. Evo 1 had a very long safety statement, and Evo 2 we worked with the safety team throughout. I think it's a lot of model developers don't even do anything. So our safety evaluations not only were comprehensive. I think they're the most comprehensive safety evaluations for an AI system that have ever been put out. Actually,
I guess I'm asking not just what you're doing, but as the technical waters rise. You mentioned other labs are not doing what we do, but that's the concern. What do we do as a society? I'm asking. This is not to be answered now, of course, but to be thought about.
A couple of extra things to say on that. In the general problem of possible effects, I think it's important to realize that these are models that we can run on computers and those generate long sequences, but you need labs, right? You need large, well-resourced labs. It's not something that somebody can generate on their computer and go into their garage and do this. So that gives us a little comfort. The other thing I think to think about is that with respect to maybe several things of concern, bad actors, and accidents, and then maybe ethical uses like designer humans, which I think are all separate things to think about. I know the ethical uses, that's kind of a common general sorts of things issues that we already have. For the accidents, that's also, I think, the accidents, I think that's also a general question about all applications of AI, which we really need to continue to think about and we haven't solved yet. For the bad actors, I think the question is, there are regions of the genomic space which we don't want to go. I think we need to go there for research. So we need to, have people have access to unrestricted models for research purposes, but then for other generally released models, we need to have some way to say don't go there. And I think that's a little bit easier than with language models, because if you want to have restrict language models, there's a lot of ways that you can produce negative language output. I think with respect to these DNA models, which will be coupled with language interfaces, so please design me a pathogenic virus, etc. I think it'll be a little bit, I expect it might be a little bit easier to tell the model don't go there, because if we can anticipate the pathogenic region, then we can build it into the model or restrict it in various ways. Even if you can jailbreak a language model, then it may be more difficult to jailbreak a DNA model in this way.
So as a follow-up to this, and I don't expect you to have solved this problem because we haven't solved it for language models either. But and we've debated, the value of fully open so that people can see what's happening. But now your models, they're already out there now. So it's escaped, right? Like you can't even stop it if you wanted to, the Evo 2 that you've put out there open source. Is there a point where we would come back and say, oh, maybe that wasn't a good idea because somebody could do something with it? And what might we do in the future? Do we have we thought through what we might change? I mean, I understand the theoretical of, if we have a language model, then we'll put language model guardrails on it. But like, how much thought has gone into that?
Yeah, I think it's a great question. One thing that we've been thinking about, and that is actually fundamentally different from a DNA language model as compared to like a natural language model, is the model produces DNA sequence, right? And it spits out a DNA sequence, and I developer of the model have no idea if that DNA sequence is right. We can test it, but we actually don't know if this DNA sequence is correct until it's been synthesized and turned from the world of bits to atoms, so to speak. And so that is also one level in which the models actually differ. If you have a model that's spewing like toxic language or graphic content, a human will immediately be able to know that that's bad and problematic. Here with biology, you need to actually make the DNA. So there's actually been a lot of, and we think these are good ideas too, not only on the modeling side, but actually adding guardrails on the DNA synthesis side. And we actually think that for now, potentially it might actually be safer to open source the models than to make them closed, because if you open source the model, you actually enable researchers to understand like the range of outputs that these models can produce, and then maybe actually use this range of outputs from the model as part of DNA synthesis screening protocols. I think Eric had a great paper on this for protein language models. But if you have an open model that people can experiment with, that can identify the like where the model is now potentially enabling researchers to generate new harmful things, then that model can just be directly put into synthesis screening efforts, for example. But it's definitely an interesting it's an interesting question. Like for Evo2, we don't even train the model on human viral sequences. So we open source the model, but we exclude everything, any virus that not only infects humans, but any eukaryotes. So this is like anything from yeast up through all the vertebrates. We actually empirically show that these data exclusions work too. And then we open source the models. But it's an interesting.
I mean, I appreciate that. Rely on, you still have to synthesize it, and Eric's talked about how to work with those companies to avoid it, but it still seems like the risk is not random person does this, it's the disgruntled postdoc is what you called it. And then I also wonder what happens when that technology no longer needs to be, that I have to send it to a company and I'm able to kind of do it on the bench. Anyways, again, I don't expect you to solve it. I'm just kind of curious what the thinking is on it. And then the last one I had is you talked about the programming language as a way to like deal with this. I'm curious, what would it be? We saw Ure's kind of chatbot for accessing their kind of model. Is that something that we would expect for science to be able to play with this, or is that just too much further out from how you're thinking about it? Because chatbot versus programming language feel like very far away in terms of what you're thinking of the use to me.
Yeah, I do think a lot of the work around agents and natural language interfaces could be useful. Biomni could, for example, Ure stuff could wrap Evo and interact with tool use. There are aspects of biological sequences that are biological design in particular that are probably less well suited for a natural language interface for now, which is why we kind of think about it as a programming language. But it is definitely one interesting interface, and we probably could wrap the programming language itself with a natural language interface too. But I think we can also be pretty creative with and develop the higher level system in a way that's more tailored to specific problems in biological design.
I'll just add a comment there. I think maybe you need a new interface paradigm. That's
think,
not a
yeah.
chatbot or a program language.
No, yeah. As we've been playing around with this, like it's a lot of it actually presents like a lot of interesting use cases that are not similar to the chatbot paradigm. A lot of these design tools, for example, take a long time to run, and it's made but there's a lot of interesting ways to make these more human accessible that go beyond the current paradigms.
Yeah, we have time for one audience question.
Thank you. Very impressive work. Thank you. So as you alluded, this kind of models needs a massive amount of data, different tier of the data feeding to the model. I'm wondering, what's your thought on the synthetic data for the different layer of data, let's say for the genomics, proteomics, or so forth?
Yeah. Actually, in genomics, we're in this like kind of one region of biology where we're not data limited, actually. And for Evo 2, we were actively discarding tokens, and we couldn't even complete a single training epoch, over our pre-training data set. So we're potentially not data limited in the in the data that gets sampled from evolution. But if you wanted to use the model to design DNA sequences that have never been sampled by evolution before, or these highly artificial systems, potentially that could benefit from collecting new data in the lab or generating synthetic data. In biology, it's somewhat nice in that certain areas of biology are amenable to these very high throughput experiments where you can collect millions of data points actually fairly cheaply. So there this is not, it's just new data collection. We've also thought about like using the models to guide ways to annotate the genomes to find new material to train, but it is an interesting.
I think it would be interesting to make a graph similar to the one that you saw with the all knowledge of humans versus the asymptotic level that we're getting by training, and we're almost there for the existing or potential genetic sequences. I don't think we're probably close, at least for the potential.
Yeah,
there's a lot of genetic data out there available.
Even a 40 billion parameter model on 9 trillion tokens was not like plateauing or overfitting at all. And our perplexities are still pretty high. I don't know. I think there's a lot of room to go with the existing data we have. And I think it is true that like the amount of DNA sequence data, just the volume of it, a lot of it is really repetitive, does actually outpace data generation on the internet. So it's not like this data, the existing data that we sample from nature will I don't know, run out anytime soon.
All right, let's give a round of applause to our final team.